Bismillahirrahmanirrahim. Now, this is the part 2 of Plasma Proteins, Chemistry and Functions, textbook by A.N. Chatterjee, pages 118 to 121. So these are the exclusive pages of 120 and 121. 118 and 119 I have already explained you in my previous lecture. Now let's start with this. So first of all, you must have the theme of this plasma proteins, chemistry and functions. Now actually we are going to, I'm going to explain the different types of these plasma proteins. So there are total 5 plasma proteins. Number 1 is the alpha globulins. Number 2 is the alpha 2 globulins. Number 3 is beta globulins. Number uh, 4 is beta 2 microglobulin, and number 5 is gamma globulins. Now we are going to have the discussion of all these 5 plasma proteins. So alpha globulins, they are further divided into 4 things. Number 1 is the alpha 1 acid glycoprotein. Number 2 is alpha 1 fetoglobulin, which is also called alpha 1 dash fetoprotein. Number 3 is alpha 1 globulins inhibitors. And number 4 is alpha 1 antitrypsin, which is also called as alpha 1 dash AT. The second kind of globulin is alpha 2 globulin. There are 2 types. Number 1 is the ceruloplasmin. Number 2 is the hepatoglobin. Then there is a third kind of globulin that is known as beta globulins. It is further having divided into further classes. Number 1 is the beta lipoproteins, which is abbreviated as LDL. Number 2 is the transferrin. Number 2 is the transferrin. Number 3 is the C-reactive protein. And number 4 is the hemopexin. Number 5 is the C1q. Then we come to fourth kind, that is known as beta microglobulin. It has no further classification. And the last one is the gamma globulins. Now, in this lecture, I'm going to give you some explanations of all these plasma proteins and their location and their functions and so on. Let's move to slide number 2. That is the we need to discuss the nature of different fractions of globulins. So first kind is the alpha globulin. Okay, so let's go to the first kind, which is known as alpha globulin. So these are actually known as glycoproteins, and they are further classified into alpha 1 and alpha 2 depending on the electrophoretic mobility. And of course, it is the clinically it is very important, alpha 1 globulins. Now this is also called, the first kind that is known as alpha 1 acid glycoprotein. Now this is also called orosomucoid. Okay, this is also known as orosomucoid. Now the concentration of this in normal plasma is about 0.6 to 1.4 grams per liter. So average is equal to 0.9. Or the carbohydrate content in this is about 41%. So it is, orosomucoid is considered to be a reliable indicator of acute inflammation. Now this acute inflammation can be anywhere. Okay. स्लाइड नंबर 2 में हमने एक और टर्मिनोलॉजी यूज की थी, that is known as electrophoretic mobility. Now what does it mean? This is actually defined as the rate of migration centimeters per second per unit field. So strength is always in volts per centimeter. Mu is equal to, तो फॉर्मूला इसका ये आता है, mu is equal to q over 6 pi r meter. So this is the formula by which we can calculate rate of migration per unit field, where mu is the electrophoretic mobility, q is the net charge on the ion, r is the radius ionic radius of the solute, and beta is the viscosity of the medium. So by this we can, so alpha globulins, these are actually glycoproteins, ठीक है, और इनको हमने जो further classification की है, that is as per the electrophoretic mobility, कि इनका जो rate of migration per unit field है वो कितना है, उसके हिसाब से we have further divided into alpha 1 and alpha 2. Next we come to this next slide, orosomucoid. And this is very important, orosomucoid, and its abbreviation is ORM. That is the acute phase, actually, acute phase protein, plasma alpha globulin, glycoprotein, and it is actually modulated by two polymorphic genes. Now, what is meant by polymorphic? What is meant by polymorphic genes? So polymorphic genes, actually, when one or more than allele occupies the gene's locus within a population. Now, in this diagram, I will explain you what is meant by the polymorphic genes. Now, it is the gene area, ये one locus है, ठीक है, ये इसमें red gene allele है, it is here blue, ठीक है, ये homologous chromosome है, ठीक है, homologous chromosome में एक blue allele है और एक red है, और तो area है, इसे कहते हैं gene locus. Locus is actually the plural of loci. This is actually a specific fixed position on a chromosome where a particular gene or genetic marker is located. Now the genetic marker over here is red and the genetic marker over here is blue. So it is okay. So allele, gene alleles हम इन्हें कहते हैं, which is you know that allele is a variant of gene, और each gene each cell has two alleles, ठीक है, इसके दो alleles हैं, one per parent, एक parent से एक allele, दूसरे से दूसरा allele, for which gene one dominant and one recessive. और जो जीन इनमें डोमिनेंट होगी वो अपीयर हो जाएगी नेक्स्ट जनरेशन में, और जो रिसेसिव होगी वो नहीं, लेकिन कंट्रीब्यूटेड बाय वन you know per parent. So this orosomucoid is synthesized primarily, okay, synthesized primarily in hepatocytes. Hepatocytes कहते हैं liver cells को, and has normal plasma concentration between 0.6 to 1.2 milligram per milliliter. That is 1 to 3% plasma protein is made up of this. Next, we come to these functions of orosomucoid. So orosomucoid actually binds the hormone, which is known as progesterone, and function as transport protein for this hormone. So probably this serves to carry needed carbohydrate constituents to the sites of the tissue repair following injury. So since it has a high carbohydrate content, 41%. So this is the function of that. It is the carbohydrate, to provide carbohydrate constituents where the tissues are injured and they need repairing. Okay. Now, next is slide. This is clinical importance of this orosomucoid. अगर increase हो जाए इसका level, so consistently rises in acute and chronic inflammatory diseases. इसका increase होता है level cirrhosis of the liver में, जो cancer है liver का, and in many malignant conditions, or जितनी भी dangerous or cancerian conditions. और decrease होता है low concentration, abnormally low occurs in generalized hypoproteinemic conditions. जब आपके पास बहुत ज़्यादा जो है proteinemia हो जाए उस conditions में, या in other words we can say that diseases related to liver. इसके अलावा आ जाता है आपका cachexia or malnutrition and nephrotic syndrome. So these are the some diseases in which this is very their levels are increase or decrease. Now we come to slide. This is number 2 kind, that is known as alpha 1 fetoglobulin, or in other words, it is also known as alpha fetoprotein. So it is present in high concentration in fetal blood during mid-pregnancy, and normal adult blood has less than 1 microgram per 100 milliliter, but may be increased during the pregnancy. Next, we go to slide number 8, which is explaining the third type, which is known as alpha-1 globulins inhibitors. So some of the alpha-1 globulins act as inhibitors of coagulation, and it also inhibited some digestive enzymes like trypsin and chymotrypsin. Okay. So next we go to the fourth kind, which is known as antitrypsin, or abbreviated as alpha-1 AT. So it is alpha-1 antiproteinase. Okay? Molecular weight approximately 45,000 to 54,000. Isoelectric point जो है इसका, that is 4. Normal value in adult it's 2 to 4 grams per liter, approximately to 90 milligram per milliliter. And this protein is highly polymorphic, that the multiple forms can be separated by electrophoresis, and it is synthesized by liver, and its principles and it is the principal protease inhibitor of human plasma. So this is very important, right? Next, we go to second kind of globulin, which is known as alpha-2 globulins, and you know it has 2 kinds. Number 1 is the ceruloplasmin, and number 2 is the hepatoglobin. So let's go to ceruloplasmin. It is a copper-containing alpha-2 globulin, a glycoprotein with enzyme activities. So इसकी enzymatic activities है, molecular weight इसका 151,000 है, और इसकी 8 sites हैं for binding copper, about 8 atoms of copper per molecule, and half as cuprous and half as cupric. It carries 0.35% copper by weight, and normal plasma contains approximately 30 milligram per 100 ml, and about 75 to 100 micrograms of copper will be present in 100 milliliter of plasma. So it is an enzyme activities, for example, copper oxidase, histaminase, and ferrous oxidase. Next, we go to the next slide. That is known as second kind, which is known as hepatoglobin. So this is another alpha-2 globulin present in plasma of clinical importance. So it is composed of 2 kinds of polypeptide chains, 2 alpha chains, possibly 3, and only 1 form of beta chain. So site of formation है, normally it is synthesized principally in liver by hepatocytes, and to a very small extent in the cells of RE cells. Now the question is, what is meant by RE cells? So RE cells is actually known as reticuloendothelial cells. So these are actually the group of cells lining the sinusoids of liver, spleen, and bone marrow. Sinusoids are actually irregular shape of blood vessels. Next, we go to slide number 12, which is actually the third kind of globulin, which is known as beta globulin. And you know it has 2 forms. Number 1 is the beta lipoprotein. So let's explain the first form. Actually, it has 5 forms. Okay, so let's go to first, which is lipoproteins, and it is actually known as LDL. So lipoprotein, you know, is a biochemical assembly whose primary purpose is to transport hydrophobic lipids. Okay, hydrophobic lipids. So hydrophobic lipids, and these are also known as fat molecules in water, as in blood plasma or other extracellular fluids. अच्छा, इनकी सिंगल लेयर फॉस्फोलिपिड्स हैं, एंड कोलेस्ट्रॉल, इसके जो सिंगल लेयर है इसमें कोलेस्ट्रॉल आउटर शेल है, और उस कोलेस्ट्रॉल का जो हाइड्रोफिलिक पोर्शन है दैट इज ओरिएंटेड टुवर्ड्स द सराउंडिंग वाटर एंड द लिपोफिलिक प्रोटीन पोर्शन जो है दैट इज oriented inwards towards the lipid molecules within the particles. So thus the complex serves to actually emulsifies the fats in extracellular fluids. Next we come to transferrin. So it is non-heme iron containing protein and formerly used to called as siderophilin. It exists in plasma as beta globulin, which is a glycoprotein with molecular weight 70,000 approximately, and it is a true carrier of iron. So protein part is apotransferrin. This is a single chain polypeptide about the size of albumin and can bind two atoms of iron to form transferrin. So its concentration is normal plasma is 1.8 to 2.7 gram per liter. Average is 2.3 grams per liter. So it has the capacity to bind 3.4 milligram of iron per liter or 1.46 milligram of ferric per grams of transferrin. And normally transferrin is about 33% saturated with iron. So this is actually the composition of the transferrin. इसकी site of synthesis जो है is largely in liver, and there is also evidence of its synthesis in bone marrow, spleen, and lymph nodes, and perhaps the lymphocytes. Next, we go to slide number 15. So, what is the function of transferrin? The function of transferrin is to transport of iron between intestine and site of synthesis of hemoglobin Hb. Okay. And it has also been seen that unsaturated transferrin has a bacteriostatic function. Now, bacteriostatic function is actually, it is known as biological chemical agent that actually stops bacteria from reproducing. Okay. And this is which is attributed to sequestration of iron required by microorganisms. So sequestration of sequestration means actually the net removal of carbon dioxide from the atmosphere by plants and microorganisms, and its storage in vegetative biomass in soils. So it has bacteriostatic function, which is attributed to sequestration of iron required by fix करते हैं ये इसको iron को. Third, इसकी kind आ जाती है C-reactive protein. It is also a beta globulin. It precipitates with group C polysaccharide of pneumococci in the presence of calcium ions. Hence, it is named as C-reactive protein. So, two electrophoretic forms actually appear on immunoelectrophoresis जब हम separation करते हैं. CRP alone in the gamma band and CRP complex. Next, we come to the fourth type, that is the hemopexin. So, hemopexin, it is a beta globulin and migrates electrophoretically in the globulin region in the electrophotogram. So, electrophotogram in the globulin region in the electrophotogram, molecular weight is 57,000 to 80,000. Normal value in adults 6.5 to 1 gram per liter. The level of hemopexin is very low at birth, but reaches adult values within the first year of life, and it is normally synthesized by parenchymal cells of the liver. So what is meant by migrates electrophoretically in the globulin? So this is actually the migration of charged colloidal particles or molecules through a stationary medium under the influence of an applied electric field. So hemopexin जो है, hemopexin इसको actually this is a plasma protein with highest binding affinity to heme. It is mainly expressed in the liver. Then we come to next fifth type, which is known as complement CIQ. So complement is a collective term for several plasma proteins that are precursors for certain active proteins in circulating in blood. So these proteins participate in immune reaction in the body. After the formation of immune complexes, C1q is the first complement factor that is bound. Okay, so the binding takes place at the Fc of constant part of the uh, IgG or IgM molecule, and the binding triggers have classical complementary pathway. So first you should know that IgG or IgM is actually known as immunoglobulin antibody. Okay, so these are actually the classical complement pathways, and it is one of the three pathways which actually activate the complement system, which is the part of immune system. And actually it is initiated by antibody complexes with the antibody isotypes of Ig or IgM. Okay, so this is actually the explanation. The fourth kind of globulin is beta 2 microglobulins. It is a low molecular weight peptide containing 100 amino acid residues and is executed in urine. It is present in urine to the extent of only 0.01 milligram per 100 milliliter, and it has also close structural resemblance to immunoglobulins. Next is the last one is the gamma globulins, and these are immunoglobulins having antibody activity. So this is about this lecture, and I have also uploaded the additional resource, a YouTube link, and you watch the video very carefully, and then in that video you will have some information about the structure of globulins and two important chains हैं, और the two important chains हैं उसमें one chain एक variable chain है और एक constant chain है. तो जो light chain particles parts को आपने observe करना है, heavy chains को करना है, फिर आपने variable regions के जो role हैं वो देखने हैं, constant regions के. इसके अलावा आपने ये देखना है कि what type of bonds are present which are actually connecting these two chains, और फिर आपने डिटेल में जो मैंने इंस्ट्रक्शंस उस असाइनमेंट में लिखी हैं उसके हिसाब से आपने एक्सप्लेन करना है टाइप्स ऑफ एंटीबॉडीज. होप कि दिस lecture is understood very clearly. If you further need my guidance or if you have queries, please don't hesitate to contact me. Okay, so that is the end of this